Meiosis is a type of cell division that produces gametes. Gametes are male or female sexual reproduction cells that fuse with other cells during fertilization. When meiosis takes place, the newly formed cells will contain half the number of chromosomes as other cells in the body. Eukaryotic body cells have a diploid nucleus containing 2 copies of each chromosome. You might know that humans have a diploid number of 46 chromosomes in 23 pairs. During sexual reproduction, 2 gametes fuse together. Each gamete must contain only one of each chromosome pair, so the offspring receives the correct number of chromosomes. So each gamete contains half the diploid number of chromosomes. This is called a haploid number. The reduction from a diploid number to a haploid number happens during meiosis, which is why it's known as a reduction division. I'll now go through the process of meiosis. The first step is interphase 1. Here chromosomes are replicated and consist of 2 identical sister chromatids joined in the centromere. In the prophase 1, the chromosomes supercoil and the homologous pairs of chromosomes line up side by side. Exchange of genetic material between pairs can occur at the crossing over point. New combinations of alleles are formed and genetic variety among the resulting gametes increases. Final step is the formation of spindle microtubulus and the breakdown of the nuclear envelope. During metaphase, chromosomes line up in the center of the cell, across the equator, being attached by centromeres. The alignment of chromosomes is random. The microtubulus now contract towards opposite poles. The pairs of sister chromatids remain together, but the homologous pairs are separated. This is where the chromosomes number changes from diploid to haploid. Spindles break down and a new nuclear envelope forms around each new nucleus. Cytokinesis follows and the cell splits into 2 cells, each containing only 1 chromosome of each homologous pair. Prophase 2 is quite similar to the first one. Here new spindle microtubuli start to form in each of the 2 new cells. The chromosome recoil and the nuclear envelope begins to break down. During metaphase 2, the nuclear envelope is broken down and individual chromosomes line up. Spindle fibers from opposite ends of the cells attach to each chromatid. Sister chromatids are separated as the centromere splits and spindle fibers pull the chromatids to opposite ends of the cell. Telophase is the last step. Now nuclear envelope begins to develop around the 4 new haploid nuclei, and the chromosomes now uncoil.